Hello. So a very good morning to all of you. So I welcome our guest from Japan, Professor Masaharu Seno, our guest from the United Kingdom, Professor Chittaranjan Chaudhary, and our very own Dr. Sushanta Rai Chaudhary, our former Vice Chancellor, Dr. Ashok Ranjan Thakur, our mentor, Dr. Arunendranath Lahiri Majumdar, and of course, our registrar, Mr. Sumanto Basu. I'm sorry. Okay. So today, we have all gathered here for the international seminar on transformative advances in cancer biology, bridging molecular understanding and biomedical innovation. So we'll discuss that here. As per our tradition of Sister Nivedita University, we inaugurate any program by loving the nature, by watering the plant. So I would request all the eminent guests and the faculties to please come forward for the watering of plant. So we will start with the welcome address by our registrar, Mr. Sumanto Basu. So I would request Registrar Sir to please come on the dais.
So I'm a man of few words, so I'll stick to that. I have a very small welcome for all of you. Respected dignitaries, distinguished guests, esteemed faculty members, researchers, and my dear students. It is my honor indeed to stand at this international seminar, which is, as you can see, is a very important topic, quote-unquote, transformating advances in cancer biology, bridging molecular understanding and biomedical innovation. Now, while today's focus is on cancer biology, this event symbolizes something much greater, the growing spirit of scientific exploration, collaboration, and innovation at Sister Nivedita University. Over the past few years, our university has witnessed a remarkable evolution in its research ecosystem, from molecular biology to computational biotechnology, from microbiological research to translational health sciences. Each department has contributed to the larger vision of making science not only a pursuit of knowledge, but also a tool for human welfare. The laboratories of SNU have become spaces where curiosity meets creativity and where young minds are encouraged to think beyond the boundaries of textbooks. Seminars like this are vital because they bridge the gap between classroom learning and real-world scientific innovation. They allow students and scholars to interact with pioneering researchers from across the world, exchange ideas, and gain insights into the latest technological and conceptual advances. More importantly, such gatherings reminds us that science is not a solitary endeavor. It is a collective journey where every discussion, every experiment, and every collaboration brings us one step closer to understanding life itself. In today's interconnected world, the field of biosciences, or what we often refer to as the biofield, has grown tremendously. From genetic engineering to stem cell research, from biomaker discovery to regenerative medicine, every branch of biological science contributes to improving human health, sustainability, and quality of life. The rapid progress that we see has further expanded our ability to tackle diseases, design sustainable solutions, and understand the complex mechanisms of living systems. Now, at its core, the purpose of research is not merely to publish findings, but to serve humanity, to heal, to protect, and to create a better future. Whether it is through understanding the intricacies of cancer cells or developing sustainable health technologies, every contribution adds value to this noble pursuit. I believe that this seminar reflects the essence of what Sister Nivedita University stands for: education that inspires, research that innovates, and science that serves. Let us take inspiration from today's discussions to continue building a culture of inquiry, compassion, and excellence in the life sciences. To conclude, let us remember that progress in biotechnology and biomedical sciences is not only about technological advances, it is about transforming lives. And with institutions like ours leading the way, we are confident that the future of bioscience research will continue to illuminate new paths beyond toward human welfare and global well-being. With these few words, I also would like to congratulate everyone involved who brought about bringing us together to have this seminar here. Thank you and have a great day.
Thank you, Registrar Sir. So next, I would request our former Vice Chancellor and mentor, Dr. Ashok Ranjan Thakur, to please come up on dais. So Sir will be addressing the House.
Good morning. One of the advantages or disadvantages of getting old is that people think that you need to get some space and some time. But today we have two excellent speakers, followed by two younger colleagues, who would be speaking on a subject, as our Honorable Registrar Sir has already mentioned to you, which is very vital for understanding, and there have been eons of developments which has taken place in these two areas. At a personal level, I got to meet Professor Seno almost at the start of the program with the Okayama University, and we have been honored with the kind of cooperation and collaboration that has occurred between SNU and the Okayama University in this particular area. There have been great efforts both by the faculties and the younger students through our exchange programs and some understanding of the collaborative program. But what I think all of us must look forward to now is getting more concrete understanding and the results of such collaboration. Because everything will hinge upon how we adapt ourselves to the greater understanding that is fostered in this realm of science and technology, and make ourselves worthy warriors of the same. About Professor Chittaranjan Chaudhary, Professor Doctor, I had the honor of being associated somewhat with him for a pretty long time. It almost spans over more than a decade and a half, I would think. And he has been one of the indefatigable kind of warriors in this area of oral cancer biology. And over the years, he has given tremendous amount of input. But what I have always liked about Professor Chowdhury is that he always lends out his helping hand to any young group that seeks to develop their own knowledge and expertise in the area. With a very free mind, with a very free understanding, and an extremely collaborative mindset. So, on a personal level, and both on behalf of Sister Nivedita University, with which in this particular university I started the last part of my leg of my journey in academics, I welcome them wholeheartedly, and I would expect our students to follow these lectures and ask questions. You know, it's one of the most disappointing note when an eminent lecturer delivers his or her understanding of the subject and then gets a stare-back look without any questions being formed in the minds of the younger people. You can only enliven the process by asking questions. No matter, there are no stupid questions. There are only stupid silences. So let us not get into that mode. Let us be very interactive. And I'm absolutely sure you'll be thrilled when Professor Seno or Professor Chowdhury speaks on their own areas of research. With these words, I thank you all for your kind patience. Thank you.
Thank you, sir. So next I would like to request our Dean of the School of Health Science and Translational Research, Professor Amit Sharkar, to please take the dais for his address. Sir.
Honorable Professor Thakur, Professor Sino, Professor Chittaranjan Chaudhary, Professor Lahiri Majumdar, Professor Rai Chaudhary, my eminent faculty members, and my dear students. A very good morning to all of you. It is really my immense pleasure to welcome you all to this one-day international seminar on transformative advances in cancer biology, jointly organized by the Department of Microbiology and Department of Biotechnology of Sister Nivedita University under the School of Health Science and Translational Research. Today's theme, Bridging Molecular Understanding and Biomedical Innovation, I think it captures the very essence of contemporary cancer biology research. As we evolve, the science evolves, we are realizing that breakthrough research seldom happens in isolation. Breakthrough research happens in the intersection of different relevant disciplines. And we hope to foster that spirit of integration and innovation through this seminar. We're deeply honored to have two internationally renowned cancer biologists here today, whose work has immensely contributed to our current understanding about the disease cancer and its treatment. Professor Masaru Sino, Emeritus Professor from Okayama University, Japan, he's a pioneer in the field of cancer stem cell engineering and translational oncology. His work has significantly impacted our understanding about the development of cancer and resistance. Is undoubtedly our pleasure, sir, to have you in this seminar. Academic Dean, City of London Dental School, University of Bolton, UK. His work in tumor suppression gene alteration in association with oral cancer with respect to tobacco use has huge importance from the public health point of view. It's a pleasure, sir, to have you with us here as a distinguished speaker. I'm also happy to mention that two of our very own young faculty members also in the speakers list. Dr. Damayanti Ghosh, Assistant Professor, Allied Health Sciences. She'll be talking this afternoon on the topic titled The Curious Case of P53 Gene in Gynecological Cancer. You also have Dr. Shani Dasgupta from Department of Microbiology, Assistant Professor. She'll be talking on the topic titled The Unsung Architect of Cancer, Why Vascular Cells Matter. Their contribution is very important for us because it reflects the growing research ecosystem of Sister Nivedita University and our deep commitment to strengthen biological research in this university. Apart from that, we'll have in this seminar two eminent cancer biologists and emeritus professors from Kolkata. Professor Raichaudhuri, Sushant Raichaudhuri is here. We'll also have with us in the afternoon Professor Tanya Das and Professor Gauri Shankar Shah. We're well aware about their immense contribution in this field of cancer biology. But most importantly to me, I would say that all these three personalities have directly or indirectly influenced academic journeys of most of the faculty members present here. So it's indeed our pleasure to have them here as the session chairs. Dear students, I would say you should consider this as an opportunity. Opportunity to listen, opportunity to ask questions, as Professor Thakur has mentioned, opportunity to engage yourself in meaningful discussion. The ideas that will be exchanged here indeed will spark the curiosity in your mind. This type of event may shape your scientific identity, can help your career to get in the right direction. Before I end, let us remind ourselves that each and every advancement in cancer biology ultimately leads to one single goal, that is lessening the suffering of the patient and forming a healthier world. I sincerely hope that the discussion that will take place here in this seminar will take us forward, one step forward towards that vision. I'd like to congratulate and thank the organizing team for making this arrangement at a very short notice, the HODs of Department of Microbiology and Biotechnology, the program coordinator, Dr. Siddhartha Datto, and his fellow colleagues, especially Dr. Sudipta Shah, Dr. Anindita Banerjee, Dr. Shurjit Singh, Dr. Rajat Pal, and others, as well as the staff members and the students. Once again, I would like to welcome you all again to this international seminar with the hope that there will be rich scientific discussion and exchange of ideas. Thank you. Thank you very much.
Thank you, sir. So now we will get into the scientific sessions. So the first distinguished talk. So I would request the session chair, Professor Sushanta Roy Chaudhary, ICMR Emeritus Scientist at CSIR Indian Institute of Chemical Biology, to please take the dais and introduce the speaker.
Good morning to all of you. Distinguished scientists and academic leaderships present in the front row, my many good friends and faculties of SNU, and also our beloved students. I think, whenever I get an invitation to come to SNU, I feel that, I'm coming to my own alma mater. We have been, associated with SNU with so many things, so many conferences, so many academic discussions that I always really enjoy this opportunity to the core of my heart. SNU is actually a center of excellence for academic activities in many, many different ways, and of course, in biomedical science, very strongly. It's really my great honor and pleasure to introduce today's distinguished speaker, Professor Seno. But as you can see, which is also very heartening to me, all speakers of these symposiums are somehow, associated with me. Of course, Professor Chaudhary, Chittaranjan Chaudhary, I know him for a long time, and actually we worked together when I was in Sharajgupta Cancer Center and Research Institute. And the other two young faculties are also in different capacities. So without going further, let me give you the formal introduction of today's distinguished speaker, Professor Masaharu Seno. He's an eminent scientist and globally recognized leader in the field of cancer stem biology, molecular biotechnology, and biomedical engineering. When I read this cancer stems biology, it's really echoes me so deeply, because I personally also very much interested in understanding this very difficult property of the cancer cells, which is now actually the center of all attraction. We believe that we are unable to conquer cancer because of these culprits that are present in the cancer, in the tumor. He's serving as a professor emeritus in Department of Cancer Stem Cell Engineering at the Graduate School of Interdisciplinary Science and Engineering in Health Sciences, Okayama University, Japan. And I learned that, this Okayama University is now, having a great association with the SNU, which is really a great heartening that, that these two great places are joining together to contribute this area of, cancer biology and other biomedical areas. Professor Seno earned his doctoral degree in engineering from Osaka University, after which he pursued advanced research across multiple domains, including gene expression, protein engineering, and regenerative medicine. Over the years, his distinguished career, he has pioneered transformative research exploring in mechanisms of cancer stemness, tumor microenvironment, and targeted therapies. That's actually reshaping the scientific understanding of cancer initiation, progress, and therapeutic decisions. He's widely known for his groundbreaking work on human-induced pluripotent stem cells. That's the key of everything, of all tissue engineering that we are looking in the future, and cancer stem cells characterization using various biotechnological and bioengineering approaches. Professor Seno has a number of numerous high-impact publications as is expected, as well as patents and book chapters, and has been moving around the world for delivering and dissipating his knowledge in this particular subject in various countries. Beyond the research excellence, he has played a pivotal role in establishing industry-academia partnership, which is very important nowadays. We really want to, translate our knowledge base to some useful entities that can help the humankind. So, by nurturing various, interdisciplinary learnings, and I think with these few words, may I invite Professor Seno to give his important talk on cancer stem cells and its engineering. Professor Seno, please.
Thank you very much for kind introduction for me. And I'm appreciate for the invitation to the such a big seminar for students and current staffs of cancer biology. And in this talk, I would like to introduce my current research initiated a decade ago. So actually, there is a long history as he introduced me because I started my career from biotechnology from Thank you very much. It's much better, I think. And so now I'm talking about the cancer stem cells, especially on advent, plasticity, and resistance today. Okay. First of all, my motivation to study the cancer stem cells, little information is available about is the laser? No. Pointer is Oh, okay. Sorry. Can I use my pointer? Okay. Thank you. But slide. Okay. I think you can put it. Yeah, okay. It's not working. Sorry. I'm sorry. It is very difficult to show the pointer here. Okay. No, this is not. This is not what I asked to you. Thank you. Sorry, pointer is not available. So I'm talking about the cancer stem cells from the point of view of advent, when it is appearing. And actually, few paper is found that is describing when the cancer cells are appearing. Many reviews or papers are describing that the initiation as the cancer cells. If you are writing cancer cells, there is cancer. But I'm talking about how normal state is shifting to malignant state. So that there should be a critical point that normal cells fell into cancer cells. Many theories are talking about mutations and viral infections trying to transform cells. But actually, the cancer patients are diagnosed. 75 to 80% of cancer patients are diagnosed as sporadic. That means that what is the reason? They don't know. So I don't deny the mutations and viral infections are the cause of cancer. But is that all? This is my question. Should be there are, that tissues are repeating regeneration during the life, so that the cancer stem cells will be appearing due to some causes to shift to the cancer stem cells. And I'm talking about point of shift from normal to malignant in this. Actually, I'm using the word of conversion, not transformation, in my story, because I want to distinguish this situation. And to explain this hypothesis, I'm using the pluripotent stem cells, and actually pluripotent stem cells are currently used as a source of regeneration therapy. Many people are talking about the microenvironment, the conditions of the proper differentiation to the mature cells, such as macrophage, blood cells, neural cells, cardiac cells, and so on. So many people are thinking that pluripotent stem cells are available to develop many phenotypes of the cells, but normal cells. But I'm thinking that the cancerous niche will be inducing the cancer stem cells from pluripotent stem cells. So once cancer stem cells are appearing, the cancer cells will be developed. This is the first idea we had. And to demonstrate this cancer stem cells appearance, we employed the iPS cells developed by Professor Yamanaka in Kyoto University. This is the mouse iPS cells derived from the transgenic mouse, of which genome has being integrated with GFP gene and puromycin resistant gene under the promotable nano gene. This means that undifferentiated fibroblasts, iPS cells are showing GFP, but if they are differentiated, GFP will go. And we used the conditioned media of various cancer cell lines to culture the iPS cells. When cultured in the presence of conditioned media for 4 weeks, the iPS cells were survived. I said survived because if iPS cells are allowed to differentiate, in 2 weeks they are differentiated and matured and starting to die, so that you cannot make culture for 4 weeks of iPS cells without conditioned media. So the important point that what is the survived cells are, you have to show the differentiation potential and self-renewal potential and tumor forming capacity of malignant tumors. These are the simple definition of the cancer stem cells. If these cells are showing the conditions, these should be cancer stem cells. If you are transplanting iPS cells into nude mice, there is a tumor, but it forms teratoma, which is benign. So that's quite different. This is not cancer, but this is cancer. So this is the model we had. And actually, it is interesting. The survived cells cultured in the adhesive cells and spheroidal morphology together with the adhesive differentiated cells are appearing. But in suspension culture, they are forming spheroid, and this means the self-renewal potential and still expressing the stem cell markers such as nanogranule. And as for the differentiation potential, actually, first of all, we did not know what kind of differentiation we can detect. But the first cancer, malignant tumor, transplanted in mice, these cells show the highly angiogenic phenotype, so that we suspected that these cells are angiogenic and differentiating into blood vessels. So when the cells are cultured in matrigel, which is a demonstration of tube formation, demonstrating the differentiation in endothelial cells. So actually, these cells form the tubes on the matrigel and differentiating into CD31 expressing cells. That means that blood vessels, endothelial cells. And interestingly, the structure shows the different type of cells. One is green fluorescent positive cells, still undifferentiated. And one is the GFP negative and CD31 positive, completely differentiated endothelial cells. And together with the yellow cells, both GFP positive and CD31 positive, but GFP negative and CD31 negative cells are mixed in this structure. So the differentiation is indicated to occurring in a discrete, not sequential procedure, but not discrete. Differentiated and undifferentiated is not discrete, not digital, I mean. There is a sequential procedure should be present in this differentiation process. Actually, in the adhesive culture, you can scan the cells from bottom to top, and there is a red part here appearing. So this is CD31 positive cells, and when analyzed, the location of the CD31 positive cells are appearing in the middle of the adhesive cells and molded sphere-forming cells. Sphere-forming cells are differentiated, and the adhesive cells are considered, we are considering that differentiated cells, so that in the middle part, the CD31 positive cells are still appearing. So we are thinking that in the same way, undifferentiated cells are differentiating negative CD31 positive cells, but by way of the middle stage of the cells. That might be perturbating indicating that there is some plasticity of the stem cell character. And actually, this is a malignant tumor derived from the converted cells. The tumor cells are sectioned to detect the malignancy. Actually, this is not benign, but malignant features are detected. So actually, these data shows that self-renewal, differentiation potential, and malignant tumor formation of the cancers, the iPS cells cultured in the conditioned media, showing the cancer stem cells properties. And we compared these survived cells with the primary culture of the tumors. One is from the tumor-derived cells. One is the metastasized tumor cells. And these cells are compared for the epigenetic study. And as a result of bisulfide sequencing of the CPG island, we found that every cancer stem cells model showing the hypomethylated CPG island when compared with the parental iPS cells. This means that the genes more than iPS cells are activated in the cancer stem cells. And when compared the number of the cells, the hypomethylated is much higher than hypermethylated. And these genes are analyzed for the KEGG pathway, and they did informatics using David informatics resources, and found that pathways in cancer, PI3 kinase act signaling pathways responsible, indicated responsible for these three cancer stem cells compared here. And in the nominated genes in the pathways, we checked the relative expression levels of these genes and found that PIK3, AP1, and PIK3R5 is upregulated. This is highly upregulated, almost 25-fold when compared with the iPS parental iPS cells. And still, in the yellow is the metastasized tumor-derived cells are still 5 to 6-fold higher in any way. So, since the PIK3R5 is a subunit of PI3 kinase gamma, this is a class 1B PI3 kinase, and there should be a catalytic domain subunit of that catalyze the kinase activities. The PI3 PIK3 CG is the catalytic subunit, so that we checked the regulation of this gene and found that the 64 upregulated when compared with the iPS cells. And the others in the tumor-derived cells and in metastasized and the survived cells are showing more than tenfold. And actually, another paper, overexpression of P3CG and P3 alpha leads to oncogenic transformation malignancy in Johnson et al. Described in 2007. And comparing the class 1A catalytic subunit PIK3CA, we checked, but no upregulation is found. So that in this case, the survived cells are showing the upregulation of PI3 kinase in class 1B. And this is the overview of the PI3 kinase signaling pathway. And there are two different pathways. One is a tyrosine kinase activation, the other is the GPCR activation. And this is class 1B and class 1, this is class 1A. So that our observation shows that GPCR activation through the class 1B PI3 kinase activating the ACT signaling for the proliferation and tumorigenicity. So the question is that what is the conditioned media we used from cancer cell lines? We can find that actually in 1980s and 1990s we found many growth factors, cytokines, chemokines, interleukins from cancer cell lines. So we can find that the conditioned medium is considerably rich in the factors. So that we thought that the iPS cells are exposed to the chronic situation. CM should be mimicking the chronic environment. So that we've hit upon a review by Brucher and Jamal showing that multi-step carcinogenesis, that is not explaining the mutation but transformation, only the chronic inflammation. Actually, inflammation and tissue damage shows the wound healing if normally treated. But if kept inflammation, this will shift to the chronic inflammation, and there will be a fibrosis, and precancer niche will be produced, and there is a chronic stress escape strategy. I'm very expressing as adaptation. Actually, progenitor and undifferentiated should be adapted to the chronic situation. And normal cell, cancer cell transition is happening. In this case, normal stem cells, progenitor cells are converting into malignant cells. And then the cancer tissues are growing and inducing the tumor angiogenesis or metastasis through the blood vessels. And then during the adaptation, actually small tumor tissues are found after 10 years, 20 years after appearance in the patients, so that during the course of time, the it is conceivable to find that some point mutations in oncogenes are reasonable, I think. So we separated the conditioned medium in two fractions. By centrifuge, you can collect the microvesicles from the conditioned media and the rest of the supernatant. And we found that both of them are potent to convert cancer the stem cells into cancer stem cells. And typically, I'm explaining that PGE2 is one sample for the soluble factor, and the other is FGF2 for the prestated exosomes. And because the FGF2 has a high affinity for extracellular matrix, if they are expressed in the recombinant technique, the cells are not showing the soluble FGF2 in the conditioned media because they are sticky to the cell surfaces and because of the high affinity to the extracellular matrix. And first of all, we use the Lucerne carcinoma, that is of which conditioned media was first found to be potent to convert iPS cells, and analyze the component of the cell membrane. And we found that this cell line is rich in the inositol phosphate. So we suspect the arachidonic acid, of which inositol phosphate is converting into the PGE2 in the inflammatory conditions. Also, inflammatory conditions shows the LTB4, but in these cases we found that LOX5 was not found to be expressed, so that we focused on this arachidonic acid leading into the PGE2 production. And actually, the Lucerne carcinoma cells are quite positive for the phospholipase A2 and COX2 and PGE2 synthesis 1 here. And this is the Western protein. So that Lucerne carcinoma is found to be positive to produce PGE2. And actually, this is the amount of PGE2 produced in Lucerne carcinoma. And as we suspected, the conditioned media is rich in PGE2 when compared with the extracellular vesicles. So we tried, iPS cells are expressing, of course, EP2 and EP4. These are the receptor, the GPCR receptor for PGE2, and if these are associated, PI3 kinases should be activated in the presence of PGE2 in PI3 kinase iPS cells. And actually, the iPS cells are responding to the PGE2 to act, phosphorylate act. So we tried the chronic exposure to the PGE2 of the PI3 iPS cells, and this is the previously obtained the cancer stem cells, but was almost the same cause. But the PGE iPS cells are survived in the presence of PGE2 during the 1 month, 28 days. So we checked the expression and self-renewal expression of cancer stem cell markers, CD44, CD133, and self-renewal potential, checking the sphere formation. And the PGE2 survival cells checked for the tumor formation, and found that these cells are forming malignant tumor when compared with the iPS cells, which is showing the benign again. The growth is much higher here. And again, we checked the differentiation potential of the PGE2 converted cells, and again, we found that CD31 positive differentiation, just the same as the first experience we found. And PIK3CG overexpression is also found in the primary culture of PGE2 survived cells. This is the cancerous cell, least lung carcinoma survived previous ones, but almost the same level, highly upregulated. And this is survived cells. And this is first obtained cancer stem cells. But all of them are showing higher upregulation of PIK3CG when compared with iPS cells. On the other hand, this is the tyrosine phosphatase counterpart of the PI3 kinase, but the expression is not so much different in these cells. Interestingly, we didn't find any significant mutations that were responsible for the transformation in these genes. And from tumor formed by the PGE2 converted cells are no more depending on the PGE2. With or without PGE2, the primary culture shows same undifferentiated GFP positive cells, and CR formation is the same here, and the tube formation as observed. So the obtained cancer stem cells are no more depending on the PGE2 presence. Means that even if the inflammation is treated, or the cells are surviving. Actually, human iPS cells we tested. In this case, we are not using PGE2, but BX species 3, pancreatic carcinoma cells. And this cell line shows is demonstrated the production of PGE2 by Bu et al. In 2011. So that we use this contagion media to convert the human iPS cells. Human iPS cells were prepared from the monocytes from blood to introducing the Yamanaka factors, and obtained iPS cells were converted into 4 weeks in the presence of conditioned media. And in this case, we used the two signal inhibitors, GSK3 and MEK inhibitors, because previously we identified these inhibitors accelerate the production of cancer stem cells. Then we found that from two different people, the monocytes are produced into the iPS cells, and they converted in the presence of conditioned media. The survived cells are checked for the activation of ACT and sphere forming ability, and differentiation into, in this case, malignant tumor formation was also checked, and the primary cultured cells are forming, and serial cells differentiation. So in this case, PGE2 is suggested to produce under the inflammatory stimulation. PGE2 are produced, and overexpressed PGE2 is keeping stimulation of PI3 kinase reaction, and if this continues chromatically, cancer stem cells will be appearing and autocrinely stimulating to growth and survival of cancer stem cells. And interestingly, we can remind the inhibitors of COX-2, aspirin. Aspirin is suggested to prevent the colon cancer, and in this case, our analogy indicates that absorption stop the PGE2 production, and PGE2 stimulation is impaired, and the chronic state is prevented, and cancer stem cells appearance will be inhibited. This is one analogy to explain that to get the cancer stem cells using aspirin. On the other hand, actually, we need to come back to the conditioned media component. I suggested the FGF2, presence of FGF2. And this is a meta-analysis data of the microarray. And these are the triple negative cancer cell lines. And we found that chronic inflammation-related factors are rich in the conditioned media expression. And we had BT549 cells, conditioned media of this cell line. We had demonstrated already the conditioned media induced the cancer stem cells, or iPS cells, and the FGF2 is since FGF2 is overexpressed, we focused on this factor. Actually, this is a very old report in 1974. Gospodarevich and Moran showed that FGF shows the morphological change in fibroblasts, 3T3 fibroblasts. Usually, in the adhesive condition, cobblestone pattern is recognized, but in the presence of FGF, the fibroblasts are converting into the crisscross pattern, that 3D spindle type shape. And this is the day for continued exposure to FGF. There's no, the spindle type is enhanced. So that at that time, the people are thinking that this kind of morphological change is a transformation of the cells. So that we checked the optimized the concentration of the FGF2 to treat the iPS cells, and we chose that as much as possible to support the growth of the iPS cells, and determined 3 microgram per ml, and this concentration was used to culture the mouse iPS cells. And 7 days, 14 days didn't show the survived cells. But after 3 weeks, 21 days of culture showed the survived cells, showing the GFP expression here. And this is before, without FGF2, differentiated cells and no more proliferating. And these cells are transplanted to the mice and showed the tumorigenicity. Sorry, before that, these cells showed sphere formation and tube formation, self-renewal, differentiation potential. And the tumor formed the malignant capacity, and showing that some KI67 and PAN cytokeratin expression in cadherin, CD44, CD24, and also cancer stem cell markers, CD133 and LDH, so that we again, we demonstrated that FGF2 over chronic exposure shows induced cancer stem cells from iPS cells. Actually, primary culture cells showed again the sphere formation, but interestingly, there's another population, sphere forming, but not GFP expressing. We haven't analyzed furthermore, but still they are positive for differentiation into the cancer, the CD31 positive cells. And the obtained cells, FGF2 converted cells, are showing, in this case, class 1A PI3 kinase were found upregulated in this case. And the ACT kinase is activated in these cells. And interestingly, the gelatin-coated cells dish shows the activation of focal adhesion kinase, PTK2, here. This is not observed in the suspension culture, sphere-forming conditions. So we thought that this mechanism are explained in this way. The FGF2 expression, overexpression, stimulates the FGF receptors, which is the tyrosine kinase, and in this case, class 1A PI3 kinase is activated through the SRC activation. And but after the conversion, the extracellular matrix are expressed, and integrin overexpression is already observed, so that this interaction is activating the first focal adhesion kinase, and this focal adhesion kinase activates the SRC, and PI3 kinase signaling pathway is activated. And this is FGF2 dependent, but its cancer stem cells are no more necessary to depend on the FGF2 here. By now we showed that the conditioned media and chronic exposure to the factors are potent to convert the iPS cells into the cancer stem cells. And now we demonstrated that interaction of adhesive cells and sphere-forming cells. In the presence of DAPT, this is a notch inhibitor. And CMAD means that conditioned media from the adhesive culture of cancer stem cells. So this indicated that this is a notch ligand expression. Spheroids are when spheroids are cultured in the conditioned media of adhesive culture here, only when this culture was carried out, the notch ligand expression was recognized. But DAPT, in the presence of DAPT, the expression is suppressed. So we thought that there's some adhesive cells are supporting the cancer stem cell survival. And this is actually sphere numbers are very significant. adhesive cells are conditioned media of adhesive cells, sphere forming cells show the difference of the sphere numbers in the cancer stem cells. And these sphere numbers are depending on the decreased, depending on the concentration of DAPT notch inhibitors. So that we suspected that we compared the notch ligands, 4 different notch ligands, and found that the adhesive conditioned media shows the significant level of JACT1 expression when compared to the other conditions. So that in this case, JACT1 were suspected to support the survival of cancer stem cells in the presence of adhesive cells. So we concluded that the differentiated cells are not more necessary, but necessary to support, maintain the cancer stem cells for self-renewing. So we think that this is the society of cells with hierarchy, different population in the same culture. Actually, in the development of cancer tissues, heterogeneity is very important. Nowadays, we are recognizing so that normal in the stem cell differentiation, once cancer stem cells appeared, the progenies are appearing in the same tissue, and these are the heterogeneity of the tumor tissue could be explained, and they are collaborating or cooperating to develop the cancer tissue. And actually, it is interesting to show that the cancer stem cells are stimulated with microvesicles, and the tumor formed, but these cells are differentiated from these cancer stem cells. And actually, the adipocyte markers are positive here, and the GFP positive cells are also present, and the merge pattern shows that yellow cells also. So middle stage of cells are detected in here. And also the CAF differentiation was recognized. Fibroblast specific protein and fibroblast associated protein and alpha-smooth muscle actin was recognized. And the transmembrane through the matrigel, invading matrigel, actually transmembrane was coded with matrigel and checked for the invasion, and the invaded cells are stained for the CAF markers, so that we concluded that some cancer stem cells developed from iPS cells are potent to differentiate it into CAF. This is the CAF-like cells invading matrigel here. And also, when transplanted, actually these cells are mammary gland tumor, derived breast cancer cell tumor, so derived cancer condition media treated iPS cells. Again, cancer stem cells converted into cancer stem cells, but we found that tumor-associated myoepithelial cells, when transplanted into the mammary fat in the nude mouse, actually, in this case, cytokeratin 8, smooth muscular actin, and the prolactin receptor positive were found. And the sections of the tumors are analyzed and showing that ductal carcinoma in situ here, and invasive carcinoma in 3 here. And this is a histochemistry showing HKI67 prolactin receptor, and cyclotin 8 estrogen receptor alpha beta. Beta is high, strongly stained. So these cancer stem cells are showing the development of the myoepithelial cells in mammary duct. And another differentiation, actually this is again showing that the endothelial cell differentiation, but based on this differentiation we further. Actually endothelial cell differentiation means that there should be a stages of hematopoietic stem cell, so that we checked so that the tumor cell high cancer stem cells to differentiate into hematopoietic stem cells, and the c-KIT and CD34 are expressing positive, and we suspected that the differentiation of the cancer stem cells into the hematopoietic stem cells. And this is another marker, this is the human iPS cells, so that the human antigen is detected here, but no mouse antigen is here. So that this means that no mouse cells are stained in this slide, so that it means that cancer stem cells are developing the hematopoietic stem cells. And the primary tumor and secondary tumor showed these character of the development. And once we found that the hematopoietic cells on the counterpart of the endothelial cells, we thought that we can demonstrate the blood cell differentiation, and found that CD34 positive cells and some part, small part, not all, but the enucleation is found. So red blood cells are found here. Under the condition of, this is a mimicry of hypoxia. Not really, but cobalt chloride. In the presence of cobalt chloride, the CD34 positive cells were recognized. And also enucleation were also accelerated in the primary culture cells under hypoxic conditions. And again, this is another experiment transplanted the human iPS-derived tumors, cancer stem cells into nude mice. And actually, in this case, macrophage invasion was found here. This is hemocyanin ion deposits inside. And I'm not showing that the macrophage is stained by anti-macrophage antibody, not for mouse, but for human. So that actually the first author found surprised to find that in nude mice, human macrophage are developing in the tumor. So that this means also suggesting that cancer stem cells developed from iPS cells are showing the differentiation into macrophage, such as called tumor-associated macrophage. So we are now thinking that demonstrated already the cancer stem cells from iPS cells are showing the hemangioblast developing the endothelial cells closely related should be with the tumor angiogenesis, and in the hypoxia conditions, hematopoietic precursor tumor-associated macrophage and erythroblast differentiation could be also detected. Another type, tumor-associated adipocytes, CAF, and these are myopithelial cells are also demonstrated, so that many phenotypes could be recognized under the various different condition of the tumors formed in the tissues. So we are thinking that cancer stem cells has various plasticity, changing the phenotypes and converting. This may be fluctuating in the stages of cancer stem cells and develop to the various phenotypes of the cells, forming the microenvironment of the cancer stem cells, cancer tissues. Yeah, actually, many people are showing that normal tissues are producing these kind of tumor-associated cells, but we are also demonstrating that cancer stem cells are potent to show the cancer-associated progenies to maintain the tumor tissues, showing the heterogeneity. And I showed that the PI3 kinase is the most important signal pathway to survive the cancer stem cells, and should be PI3 kinase inhibitor should be a good target drug to suppress the tumor growth. And actually, we showed that, demonstrated that the recurrent carcinoma-derived mouse iPS cells, or conditioned media converted iPS cells, treated with the two different inhibitors, EGF receptor and PI3 kinase gamma. And actually, PI3 kinase gamma inhibitor shows the significant suppression of the growth of the tumor. And also, EGF receptor inhibitor was effective. So, and actually, the cancer stem cells are very famous for showing the drug resistance, so that we show using the several cancer stem cells we developed, we checked the spectrum to assess the anti-drug properties. And these are the almost 200 anti-cancer drugs, and small number of the drugs are showing the suppression of the growth of these cells. This is primary culture and survived cells. And this lung metastasized, and also here, small number of the red bars shows the growth inhibition. And this is B16 melanoma cell condition media converted cells. Again, small number of drugs are found effective, and compared the cell and again the drugs again here, doxorubicin and daunorubicin found to be effective. And this is more than four. Some another cancer stem cells are compared, and actually the normal cells, fibroblasts and other cell lines are not affected, but these drugs and termorubicins are all effective. But the problem is that iPS cells are also suppressed. But it is suggested that the stem cell-like cells are suppressed by this kind of anti-cancer drugs. So sensitivity to termorubicin is found to induce the apoptosis, showing the ladder of DNA ladder. And the conclusions here. We have been demonstrating the conversion of normal stem cells to cancer stem cells using various conditions with conditioned media from cancer cell lines without intended mutations or introduction of foreign genes. The factors in the conditioned media could be addressed to those in inflammation. In every case, PI3 kinase, ACT, mTOR signal pathways are implied to be responsible for the initiation by epigenetics, but not only by mutation. The stemness of cancer stem cells is the origin of the various phenotypes of cancer-associated cells that could build up with a hierarchy and tumor tissue providing heterogeneity. And the developed cancer stem cells were resistant to almost all of anti-cancer agents assessed, but found sensitive to a couple of the agents. These cancer stem cells could be available to develop therapeutic strategy targeting cancer stem cells. So now we found that iPS cells could be developed from different individuals. Actually, even not patient, normal people could provide monocytes, and you can get iPS cells from the cell, each cell. And we know that how we can develop cancer stem cells so that we can develop the various kind of cancer stem cells with a different genomic background. So these cancer stem cells, bank of cancer stem cells could be personalized, lead to the personalized cancer treatment. We are expecting this strategy for the future perspective to develop the personalized cancer treatment with individual cancer stem cells. Thank you. Thank you very much for your kind attention.
Thank you. Hello. Can you hear me? Yeah. Okay. Thank you, Professor Seno. I think we can have some discussion on this, lecture that he has given. I think, as you can see, Professor Seno has put forward a fascinating hypothesis. It's a mutation-independent pathway of tumor development. Okay. So far, we heard that tumors are, have so many different mutations. But what he is showing is that that the normal stem cells in a tumor, in a normal tissue, under certain condition, okay, environmental conditions or so, they can convert it into a stem cells, which is capable of tumor formation. And not only that, those stem cells can also be differentiated into different other cells which can support the tumor, as we know the tumor microenvironment. Okay? So I think it's a very fascinating, I guess what I said, summarize the essence of your talk, right? So, it's now open to all. If any pressing questions or discussions or points that you want to discuss with him? Especially from the students.
First of all, thank you, sir. And actually, just few months back, I have read your paper, Cancer Initiation, I think so that was in late 2024 or 25 only. It was by you, the paper on cancer purification and initiation. So I've gone through that paper and I feel honored to see you in person presenting that to me. So, sir, in your slide number 69, you have shown this signal inhibitors inhibited tumor growth. So can we consider this EGFR and this as a combination therapy in slide number 69? Yeah, 69, yeah.
The previous one.
Mm-hmm. Yes. So we can see, sir, like we can consider this as a combination therapy. Yeah.
Actually, we tried to show the efficacy of the combination therapy.
Right. So, sir, I have a question. It was basically in the early slides and all. So, sir, considering that this the transformation is initiated by PGE2 and COX2, right, and logged by PI3K and AKT, would a combination therapy both upstream, if like, I combine both the upstream inducers such as COX2 inhibitor, like celoxips are present, and downstream AKT, will that combination therapy, will it work? So in the initial slides, you were telling that PGE3 and COX2, if I I take both the things to upregulate and downregulate, will that as a combination therapy, will it work, sir?
Actually, I wanted to show that the PI3 kinase inhibitor is effective.
Okay.
Because I showed that the PI3 kinase gene is upregulated.
Right.
Actually, this is a result of the combination therapy, but in this talk, I'm not focusing on the combination.
Right.
And actually, in this case, EGF receptor inhibitor appears to be effective. So now, but in this case, the combination is not so much effective. So that we need to know the device, I think the timing of the combination, so that not at the same time, but maybe there is some effective points.
Right,
yeah. But at this moment, I cannot say anything more.
Okay. Yeah. Thank you.
Sorry.
Thank you for the excellent speech, sir. I just had one question that for this iPSCs, do you need the basement membrane? How important is the niche? Is the niche important for in important stem cells? Or even in cancer stem cells, is the niche important? Like in normal stem cells, we know that the niche is important and the basement membrane is important.
Actually, niche is quite important because we are thinking that the most important point is that the chronic situation of that makes the activated and makes the progenitor cells and stem cells are activated, and these cells are exposed to the same factors or different inflammatory factors, and many genes are activated or suppressed in this situation. And if this is the chronic situation is continuous, these plastic cells are adapted to the niche, right? And finally,
Yeah, I think it's not.
It refers to the normal, so that this will be the initiation of the cancer, so that only not only niche, but the niche should be very much significant, induce cancer stem cells. I don't know if mutations or viral transformation or infections of oncology virus is at the same time occurring or not, but without these factors, the chronic exposure to the factors that I showed you will be very much effective to induce cancer stem cells, I think.
Very interesting talk, sir. So I have a question regarding the differentially methylated region. So one of the slides you showed that you have found more hypomethylated region than the hypermethylation in many of the cell types. So my question is, did you notice any transcription factor enrichment cell type specific for those hypomethylated region?
Actually, I'm sure that many transcription factors are related with the enhance or suppressing the genes related with the cancer stem cell occurring.
Thank you.
Yeah, actually, epigenetics of the CPG island and the HDAC could be activated, and why not?
Thank you, sir, for the nice talk. So I have two questions. So one is that you see that PI3 kinase AKT pathway is responsible for the transformation from stem cells to cancer stem cells, right? So what do you think that if any of the other stromal cells in the tumor niche, cancer niche, if they upregulate this pathway, they can also have like stemness property?
So
if like any other cells which have in the tumor niche, they have also showing the AKT PI3 kinase upregulated upregulations. Do you think that they can also have that stemness like properties?
Actually, we happen to find without the PI3 kinase activation, but I think there are another pathway should be. Actually, another case is responsible for another signal transduction pathway is related, is possibly present, such as JAK kinase and SMART, or some another inflammatory cytokine signaling pathways. There are so many, so we need to find out, to survey the various cases to find out if PI3 kinase is only the way or not. So that your question is still should be investigated, I think.
So my next question is that you have seen this cancer stem cells. So have you checked that how they can modulate immune cells within the tumor microenvironment?
Immune cells?
Yes.
Well, in this case, in our cases, leukemia is not tested yet, but it is an interesting way to check, one of the leukemia cell line K562 could be the condition media from that cell lines could convert the cells into the cancer stem cells or not. It would be another experiment, I think. Actually, the interesting way that might be possible would be the chronic inflammatory, the model mice with chronic inflammation should be the host of the transplantation of iPS cells. In that case, it will be interesting to check if the leukemia is occurring or not. Could be actually at this moment we haven't tested, but it will be a good the strategy to identify if the leukemia is happening or not.
Thank you so much, sir.
No more questions. I think we can call this exciting session, so fascinating observations and new findings. I think we thank Professor Seno for giving this excellent, fascinating lecture and the hypothesis. Thank you very much.
Thank you very much.
It is my privilege now to invite Professor Sushanto Roy Chaudhary to felicitate our honored speaker, Professor Masaharu Seno. May I now request Professor Ashok Ranjan Thakur to felicitate our Honorable Session Chair, Professor Sushanto Roy Chaudhary. We now invite our distinguished guest to please proceed to the VIP lounge for lunch. Our second distinguished talk will start from 1:30. There is an important announcement for the participants. There will be a QR code shown at the end of the program that you need to scan to get the e-certificate. Thank you. We will now disperse for the lunch break. The second distinguished talk will start from 1:30 p.m.
Hello.